大家
嗯，
都挺拼的。
我们可能会再多等1、2分钟，反正我们 lab 的学生都来了。Right，
对，再等一下好了。
yeah，一会儿我来介绍永祥，然后后面的话呢，你就帮忙负责一下 Q&A。
行行行，我一般
OK
周五这个时间我都是 available 的，所以就是说，
OK，
如果我反正我们俩都 coordinate，就是说，
yeah，
如果他们其他人没有人 host 的时候，对吧？
OK，
都没有什么
好
问题。
，OK，我再发一点到别的。We are gonna wait for 2 more minutes, and we can go ahead start. 我们再等2分钟，然后就开始。哎，永祥，你要play 一下看看，现在还不是 full screen。Okay, that's good. Yeah, right. 这回我们可以听到更多一点的数据。上一次6月在 Endo meeting 只有一小部分。
对，time this is cell paper
Yeah,
should be data。
yeah. 这个工作好像一开始还有很多数据，他们拆开的话呢，估计另一部分数据还能再发一个不错的期刊。OK, all right. 我们先开始吧。Okay, hi everyone. Good morning to the audience in South America, North America, and good evening to the audience in China. Thank you for joining us at today's CADA Happy Hour virtual talk. We understand that it might be inconvenient for those people in China and the West Coast. And this month, we are fortunate to have two outstanding postdocs who are currently on the job market. They will present their recent published work in top journals during the Happy Hour virtual seminar. And this virtual seminar will also serve as their rehearsal for the job talk. And today, I'm very happy to introduce Dr. Yunxiang Li from Baylor College of Medicine. Dr. Li got his bachelor's degree in agriculture at Foshan University in the year 2016 in China. He then studied his PhD study in South China Agricultural University in China. And then in 2021, he joined Dr. Yongxu's lab at Baylor College of Medicine to work as a postdoc till now. His research interest in Dr. Shu's lab at Baylor is to identify the molecular targets for diagnosing the onset of obesity and postpartum depression. Dr. Lee has a very successful postdoc training with his mentor at Baylor. He got one AHA postdoc fellowship and one ADA postdoc fellowship research grant just after 2 years from 2022 to 2024. And he also received the Endo Society Travel Award in the year 2024. He presented a first oral presentation about part of his work. As a first author, he published one Cell paper this year. As co-author, he published multiple papers in top journals. Those included the Nature Metabolism, Science Advance, and so on. He's now looking for an independent position to pursue his career goals. Now, today, he will present some data from his recent Cell paper. His title for today is TRPC5, Obesity and Postpartum Depression. I'm going to stop here and let's welcome Yongxiang. Thank you.
Okay, thank you. Thank you for Yin's very kind introduction. Good morning, everyone. My name is Yongxiang Li from Dr. Ying's lab in Baylor College of Medicine. Today, I'm so happy to share for my work. The title for my presentation today is TRPC5, Obesity and Postpartum Depression. So firstly, as we know, the obesity has become a very serious health problem in the world. This slide shows the increasing obesity rates in the world from 1975 to 2016. In addition, WHO estimates about 20% adults in the world will be obese by the end of 2025. Now let me introduce a little bit more information about transgender receptor potential channels, TRPC channels. TRPC channels are memory express calcium ion channel. They can transduce sensory stimuli to the electrical signal. Basically, TRPC channels has 6 members, including TRPC1 to TRPC7. Among the 7 members, TRPC5 is abundantly expressed in the brain. But why I emphasize TRPC5 channel but not other channels? Here's the reason. Recently, our lab collaborator, Dr. Sana Fuki from the University of Cambridge, he found that 2 boys with severe obesity is the unrelated families in Europe. If we look at in the left side of the body weight curve, we can say the case 1, the boy's body weight is 150 kilograms, and the case 2, the boy's body weight is 80 kilograms when they are just 14 years old. Importantly, Dr. Sanhav found that there is a small deletion in the chromosome X, and this small deletion disrupts the gene called TRPC5. In order to investigate if rare variants of TRPC5 may be identified in other people with severe obesity, we reviewed the exome sequencing data from the genetics of obesity study. We totally identified 7 TRPC5 variants in humans with obesity. We can see the K34del T134M and totally 7 variants. So next question is whether those variants will affect the normal TRPC5 expression. So Dr. Nain, previously in our lab, she transferred those different variants into the cells and found the K34del, this point variant can significantly decrease the TRPC5 expression in the cell membranes. Besides the expression of the TRPC5, the more important thing we want to look at is if those variants will affect the normal function about the TRPC5 channel. So using extrophysiology, Dr. Yang He recorded in the wild type cells, you can see CCH could induce currents, and this current could be abolished by the TRPC5 antagonist. So based on this data, we use all the TRPC5 variants and examined and found the P34 this point mutation represent most severe loss of function. So based on this data, next we made a P34 knockout mouse model. So using this model, we can say compared with the wild-type mice, the TRPC5 P34 mutant mice developed a modest body weight gain, but significantly increased the food intake. So this data suggests that TRPC5 P34 mutant mice developed obesity and hypophagia. Based on this data, we can say loss of function TRPC5 could increase food intake. So next question, I'm wondering whether activation of TRPC5 could decrease food intake. So here I used the specific the activator for TRPC5, the BDD. The new compound was developed. It can activate the TRPC5 as well as the combination with other channels. So I injected the BDD into the mice. So from the data, we can say compared with the vehicle injection group, BDD could significantly decrease the food intake, both in chow and high adults. So next question is why the BDD could decrease the food intake. So in the brain, in the hypothalamus, the pumping neurons is associated with the negative energy balance. Specifically, in the accurate nucleus, pumping neuron could release the alpha MSH. The alpha MSH could bind the MC4 in the satiety neurons to increase the satiety, which means decrease the food intake. Meanwhile, in another study that's led by Dr. Zhang Cheng almost 10 years ago, found they used the DT and DTR strategy to ablating the pumping neurons in the acute nucleus could significantly increase the daily food intake, cause hypophagia. So my question is whether BDD could activation of a pumping neuron to decrease the food intake. Before answering this question, I need to examine if a pumping neuron expresses the TRPC5. So using staining study, we can see in the GLP labeled pumping neurons, the green color pumping neurons, they can express abundant the TRPC5, the red color. So this data answer the question, pumping neuron express abundant the TRPC5. So next, I inject the vehicle and the BDD into the wild-type mice and co-stain the pumping neuron and the C-force. From the data, we can say compared with the vehicle injection group, BDD can significantly increase the C-force in pumping neurons. Because the C-force is the marker for active neuron, the more C-force expression represents the neurons activity is much higher. So this data suggesting that BDD could activate the pumping neurons to decrease food intake. So the next question is whether inhibition of a pumping neuron activity first, the BDD cannot decrease food intake. So to examine this hypothesis, so we use the GRAD strategy in here. So let me introduce a little bit about the GRAD system. GRAD is designer receptors exclusively activated by the designer drug. So how does it work? So firstly, I will inject a virus. This virus will express ATM40GI, these receptors in the cell membrane. This receptor is a designer receptors. Then I will inject a compound with CNO. CNO is the designer drug. So CNO could combine with this receptor to decrease the neurons frame rates. Meanwhile, the neurons activity will be inhibited. So using this strategy, I inject the crate-dependent GI dried virus into the pumping mice. So from this image, we can see this matrix expression represent the pumping neurons. So after adding the CNO into the pumping neuron, we can see the pumping neuron firing rate is significantly decreased after the CNO. So using this strategy, I inject the vehicle and the BDD into the seed injection group mice. We can see BDD could significantly decrease the food intake. However, when I use the CNO to inhibition of the pumping neuron activity first, BDD cannot decrease the food intake. So this data suggests that inhibition of promising neurons could block the BDD-induced anhedonic anorexia. So because previously I showed the Christopher this partner mutation represents the most severe loss of TRPC5. So next the question is if the Christopher this mutant mice can block the BDD-induced anorexia and symbolic expression. So in this study, we used the vehicle and the BDD to inject the wild-type mice. We can see BDD can significantly decrease food intake as well as increase the C4 expression in the promising neurons, which is consistent with previous data. When I use the TRPC5 for the mutant mice and injection of the BDD, we can say in the mutant group mice, BDD cannot decrease the food intake as well as did not change the C4 expression in the promising neurons. So this data indicates that the TRPC5 crystalline mutant mice can block BDD-induced anorexia and C4 expression. So this is the metabolic phenotype about the mutant mice. Besides the metabolic phenotype, we also observe the other inhibiting the behavior change. So first is the anxiety behavior. In the anxiety behavior, we use the open field test in here to examine the anxiety behavior. In the open field test, even mice spend less time in the central area, representing the mouse anxiety level is higher. So from the data, we can say compared with the wild-type mice, the TRPC5 mutant mice significantly decrease the time spent in the central area, which means this mice inhibit anxiety. So next is the regression. To examine the regression behavior, I used the resting intruder test in here. So how to do the resting intruder test? So firstly, the resting cage, the mice will be isolated for at least 1 week previous the test. Then I will introduce male intruder in the resting cage. Then the resting the mouse will choose to attack intruder or not. From the data, we can clearly say compared with the wild-type mice, the mutant group mice significantly increase the attack duration to the intruders, which means TRPC5 mutant mice exhibit aggressive-like behavior. So next is the autism. So to test the autism, we use the social interaction test. In the social interaction test, we can say they divide in the same chamber. In the left side is the object paired side. In the right side is the mouse paired side. So the experimental mouse will choose to interact with object side or the mouse side. From the data, we can say in the wild-type mice, in the normal mouse, they prefer to interact with the mouse side, but not the object side. However, in the TRPC5 mutant group mice, they did not prefer to interact with the mouse, which means this mice developed autism. By the way, the anxiety, aggression, and autism is also the symptoms we observed in the humans. So far, I want to make the summary one. The first is the loss of function in TRPC5 causes hyperphagia and obesity. The next is activation of TRPC5 inhibits food intake in mice. BDD decreases food intake through TRPC5 in promising neurons. Last is the TRPC5 crystalline mutant mice inhibit anxiety, aggression, and autism-related behaviors. So let me move to another part. So besides the obesity we found in the T-boys, another important discovery is we found that in humans TRPC5 deficiency causes postpartum depression in the mothers. If we look at the family tree, we can see the 2 boys who carry TRPC5 deletion exhibit the severe obesity. Meanwhile, their mothers also carry TRPC5 deletion and exhibit obesity and postpartum depression. We can see this green color represents the postpartum depression, and blue represents obesity, and yellow represents the anxiety. This is pretty interesting because we can see both the two moms exhibit the postpartum depression, but not only one. So speaking of the postpartum depression, it is a serious mental health disease. Postpartum depression can affect 10 to 15% of women and impaired intelligence between the mom and their babies. Importantly, postpartum depression can occur from 4 weeks and until 1 year after birth. That means it can last a long time. But that's the one thing we need to keep in mind. When we talk about the postpartum depression, it is a little bit different from the general depression. Besides the general depression, the postpartum depression also has another two different symptoms. First is it has the permanent nuclear, and another is the anxiety symptoms. So to examine, our mutant mice developed the postpartum depression behaviors. So I designed this study. So I used the wild type of the male mice to cross with the wild type or TRPC5 mutant virgin female mice and examined the related behaviors according to this timeline. So we can see after during the lactation period, we used the abandoned behavior and triple behavior to measure the maternal care ability. And after the weaning, I used the forced swim test and the sucrose preface, the two classical tests to measure the depression-like behavior. And at the postpartum days 30, I used the open field test to examine anxiety-like behavior. Surprisingly, we can see the TRPC5 mutant dames ignore and abandon their pups after delivery. This is in the postpartum day 1. From the picture, we can see in the wild type group, the mother, the dames, and the baby and the pups sit together in the nest, which is very beautiful. The nest. However, in the mutant group of mice, we can see the dam they ignore and abandon the pups. They did not care about the pups. The pups are separated in their home cage. Also quantificate the difference between abandoned behavior and the normal behavior. We can say that's the significant difference between wild type and the mutant group of mice. So next, I also used the retrieval behavior to measure the maternal care ability between the two group of mice. So here, I like to share a short videos. And we can see in the left side is the wild type of the dames. We can retrieve the pups one by one with a short latency. However, in the right side is the TRPC5 mutant mice. They did not retrieve all the pups, and they also destroy their nest. If I stop at here, we can see if I stop at here, we can see the wild type mice, they stay in the nest with their pups and will take care of the pups. However, the mutant group of mice, they did not retrieve all the pups. Also, they destroyed their nest. So for the retrieval behaviors, the dame will retrieve the pups to their nest. From the data, we can see compared with the wild type mice, the mutant group of mice, they significantly increase the latency to retrieve the pups to the nest. After retrieving the pups, the dames will exhibit several different behaviors like the crouching, grooming, nest building, and time nest. This heat map clearly shows the difference between the wild type and the TRPC5 mutant mice. We can see the behaviors, the frequency, and the behaviors, the duration is significantly decreased with the mutant group mice. Also quantificate the 4 different behaviors. You can see the crouching, grooming, nest building, and timely nest. All of them were decreased in the mutant group mice. As I mentioned before, the postpartum depression it has 3 different symptoms. So besides the maternal behaviors, I also want to look at if those dames inhabit postpartum depression. So for the postpartum depression, I use the classical experiment, the forced swim test to examine their depression-like behavior. In the forced swim test, the mice will be put in the water for 60 minutes. I will record the last 4 minutes. If the mice have a longer inbound time, represents the mice depression level is higher. From the data, we can clearly see compared with the wild-type mice, the mutant group mice gains significant increase the inbound time represent the mutant group gains with habits postpartum depression. Meanwhile, I also use the sucrose preference test to test if the mice they showed anhedonia. Briefly, I provided two different liquids. First is the water, and another is 1% sucrose. So the mice will choose to drink the water or the sucrose. From the data, we can say compared with the wild-type mice, the mutant group mice significantly decrease the preference for the sucrose, which means the mutant group mice they showed anhedonia. So together, this data suggests that TRPC5 mutant gains exhibit postpartum depression like previous. So lastly, I also use the open field box to examine if the mice exhibit anxiety-like behavior. We can say compared with the wild-type mice, the TRPC5 mutant mice gains significantly decrease the time spent in the center, which means this TRPC5 mutant game also exhibits anxiety-like behavior. However, importantly, we did not detect any difference between the wild-type and TRPC5 virgin female mice, both in forced swimming test and the sucrose preference test. So this data suggesting that TRPC5 mutant games only show the depression-like behaviors during the lactation period, but not in the virgin. So together, I'd like to make a summary too here. Loss of function in TRPC5 causes postpartum depression in mothers, and the loss of function in TRPC5 also causes postpartum depression-like behavior in mouse things. And the last is loss of function in TRPC5 did not elicit depression in virgin female mice. So far, we know the mutant mice they developed the obesity and the postpartum depression. So what's the mechanism behind this phenomenon? So given the fact that TRPC5 is abundantly expressed in the brain and the postpartum depression is a serious mental health disease. So firstly, I look at the TRPC5 expression in the brain using the brain map website. So we can see the TRPC5 is abundant in the regions in the PVH as well as SO1 because the different region they have the same points. That's in the two different regions they reach in the oxytocin neurons. So here is the brain sections from the oxytocin T tomato report mice. You can see in the PVH region and the SO1 region they located a lot of oxytocin neurons. So why oxytocin? What's the oxytocin? Oxytocin is a hormone related to the maternal behaviors. Previously, a study that published in Nature paper reports that using the drug strategy, I have introduced this drug strategy before. Use the drug strategy to inhibition of oxytocin neurons in the PVH that can decrease the retrieval probability in the games. We can see the single injection inhibition of oxytocin neuron could decrease the retrieval probability. Meanwhile, in another study, they used the oxytocin, the compound, to injection of the virgin female mice. In the virgin female mice, after injection of oxytocin, it can elicit the retrieval behaviors. However, in the single injection group, the virgin mice did not retrieve the pups. So given the oxytocin play a critical role in the maternal behavior, I suspect if oxytocin in the PVH could express TRPC5, and if yes, what's the function of it? So to answer this question, I co-spin the oxytocin and the TRPC5 in the PVH and found most 34% oxytocin neurons will express TRPC5. So I answered this first question, oxytocin neuron could express TRPC5. So next question is, what's the function of it? So next, to investigate the contribution of TRPC5 in the oxytocin neuron to the phenotypes we observed before, we used the Kralox-P approach to delete the TRPC5 from oxytocin neurons. This image validates the TRPC5 deletion in the oxytocin neurons in the male mice So after deletion of TRPC5 from oxytocin neurons, we can say compared with the control group, the deletion group mice significantly increase the body weight as well as the fat mass and the lean mass when fed chill diet. Furthermore, we use the metabolic KT to measure the metabolic change. From the data, we can say in the food intake is a significant increase in the deletion group, and the locomotion activity is a significant decrease in the deletion group. However, the energy expenditure, there's no difference when we use the body weight as a covariate. So this data indicates that hyperphagia is a major driver to the obesity in the mice with deletion of TRPC5 from oxytocin neurons. This data also suggests that in addition to POMC neurons, oxytocin neurons is another target to regulate body weight and food intake. So importantly, when we use the oxytocin compound to inject into the mice, we can say supplementation of oxytocin to the knockout mice. It can decrease the food intake as well as decrease the body weight to the level that are comparable to the wild-type mice. So it is important because oxytocin analogs or receptor agonists could be effective in treating human obesity due to TRPC5 deficiency. So besides those metabolic data, I also repeat the anxiety and autism the data in the knockout group mice. We can say compared with the control group mice, the deletion of TRPC5 from oxytocin neurons also decrease the time spent in the central area. This means the deletion group mice develop the anxiety-like behavior. Also, in the social interaction test, you can see in the control group mice, they prefer to interact with the mouse side. However, in the deletion of group mice, they do not prefer to interact with the mouse side. So this data indicate that deletion of TRPC5 from oxytocin neurons causes anxiety and autism. So how about the female mice? I also use the control and the knockout TRPC5 from oxytocin female mice to evaluate to examine the contribution of TRPC5 to the maternal behaviors. So in terms of maternal behaviors, we can say compared with the control group mice, the deletion of TRPC5 from oxytocin neurons can increase the latency to retrieve the pulse, which is similar with our mutant group games. Meanwhile, also evaluate the maternal care ability. We can say compared with the control group mice, the knockout TRPC5 from oxytocin neurons also impaired the maternal care ability, including decreased approaching, grooming, nest building, and timely nest. The most important is deletion of TRPC5 from oxytocin neurons also causes postpartum depression. As shown in the data, we can say in the fostering test, the deletion of TRPC5 can significantly increase the embryo time in the liver test. Also, deletion of TRPC5 from oxytocin neurons also decrease the preference to the sucrose, which means this knockout deletion of group mice also develop the anhedonia. So far, we already know the mutant group mice and the deletion of TRPC5 from oxytocin neurons is all loss of function TRPC5. So next question is whether gain of function TRPC5 in oxytocin neurons could improve those metabolic or behavioral deficits in the mutation group mice. So to address this question, we restored of TRPC5 in PBH oxytocin neurons. So firstly, we inject a control AAV in the left side and AAV-DL TRPC5 in the right side using a same mouse. This mouse is TRPC5 crystalline oxytocin created tomato mice. The AAV-DL TRPC5, this virus is crate dependent, which means this virus only expressed in the crate expressed neurons, which means the oxytocin crate neurons. From the image, we can see in the control AAV injection group, oxytocin neuron express very minimal the TRPC5, So because each mice is a mutant group mice, so TRPC5 express is very minimal in the left side. At the same time, in the right side, after injection of AVDL TRPC5 the virus, we can see in the red color, the oxytocin and the neurons abandon TRPC5 expression. This means the AVDL TRPC5, this virus can increase the TRPC5 expressed selectively in the oxytocin neurons. So next important question is whether restoration of TRPC5 in oxytocin neurons could rescue the cellular response of oxytocin to the TRPC5 activator. So to answer this question, I injected the BDD after the surgery after 4 weeks. So I injected the BDD into the mice which received the control AV or AVDL25. So from the data, we can say compared with the control AV injection site, AVDL25 injection site, the oxytocin neurons expressed more C-force. So this means, so this data are important because it provides evidence that restoration of TRPC5 in oxytocin neurons could rescue the cellular response of oxytocin neuron to the TRPC5 activator BDD. So after validation of this virus, so I bilaterally injected the AVDL TRPC5 into the PVH in the TRPC5 crystal delta mutant oxytocin crazy male mice. So after injection of the virus, we can say compared with the control AV injection group, AVDL TRPC5 injection group of the mice decreased the body weight both in the jaw and habitat. Meanwhile, the restoration of TRPC5 also decreased the fat mice and lean mice. Meanwhile, we also use the metabolic kg to measure the metabolic change. We can say compared with the control AV injection group, AVDL TRPC5 injection group mice significantly decrease the food intake both in the jaw phase as well as in the habitat phase. In the locomotion activity, we can see restoration of TRPC5 also increases the locomotion both in jaw and habitat, the different phases. And we also use the mass weight as the covariates and demonstrate that no difference for the energy expenditure. So besides the metabolic change, I also use the behavioral test to examine anxiety-like behavior. We can say restoration of TRPC5 could rescue the behavioral deficit in TRPC5 deficiency. After injection of TRPC5, the time spent in the center area is a significant increase in the refill box. Meanwhile, for the social interaction test, we can say the control AV injection group, which means the mutation the mice did not prefer to the mouse side. However, after restoration of TRPC5, the mice prefer to interact with the mouse side, but not the object side. So next, we also breed a cohort mice, the TRPC5 crystal del oxytocin female mice to receive the control AV or AVDL TRPC5 the virus. So after the virus surgery, we use the male mice to mate with the female mice. After 3 weeks, when the female mice delivered, I used the retrieval behavior to measure their maternal behaviors. And after weaning, we used the bone swimming test and the sucrose preference test to evaluate their postpartum depression-like behaviors. So from the data, we can say after restoration of TRPC5, the times can decrease the latency to retrieve the pulse, which means the restoration of TRPC5 could reverses the maternal behaviors deficits in TRPC5 deficiency. Meanwhile, I also evaluate other details in the behaviors, for example, the protein, as I introduced before, compared with the control AV injection group, restoration of TRPC5 in the oxytocin neurons can improve the protein grooming and the time less user times. So lastly is the postpartum depression, the other symptoms like the following test, we can say restoration of TRPC5 can significantly decrease the enable time in the following test, as well as increase the preference for the sucrose in the sucrose preference test. Last, in the open field test to examine anxiety-like behavior over restoration of TRPC5 in oxytocin neurons can significantly increase the time spent in the center area. So together, those data indicates that restoration of TRPC5 specifically in the oxytocin neurons can reverse the behavioral deficits, including the metabolic deficit as well as the behavioral deficit of TRPC5 deficiency. So here is the summary three. The first is the deletion of TRPC5 from oxytocin neurons causes obesity in mice. And the deletion of TRPC5 from oxytocin neurons can increase anxiety, impairs social ability, and causes postpartum depression in the mice. Last is restoration of TRPC5 in oxytocin neurons reduces body weight, anxiety, and postpartum depression in TRPC5, the mutant mice. So far, I've reported all the data. So here's I like to here to make a conclusion. So our data shows that the disruption of TRPC5 in humans can lead to the halting behavior. This is the novel behavior that I did not include in my slides. And in the humans also result in hyperphagia, obesity, anxiety, autism in males, and results in postpartum depression in the females. Using the mouse model, we also showed the mutant mice as well as the specific deletion of TRPC5 from oxytocin neurons, the mice. They also showed the hyperphagia, body weight gain, and postpartum depressive behaviors in the females, and reduced social interaction and cohesion in the males. So together, our data shows that TRPC5 play a pivotal role in mediating homeostasis and behavioral response that are essential for survival. So at the end, I like to thank my mentor, Dr. Yu Xu, the commander Dr. Da Yun for the guidance and help, as well as the committee members Dr. Zheng Shen and Yibing Zhou for the advices and suggestions, as well as the most importantly is thanks for Dr. Na Ying and Dr. Yang He for the sales work and ethics work, and we also special thanks for the collaborator Dr. San Fuki from the University of Cambridge and Dr. Chumi Wang from the Psychology Medicine. And thank you all. I'd like to take any questions.
Okay, thank you, Yongxiang. And I will host the Q&A section here. And first, I like to say that's very impressive work and very significant work and beautiful data. So I guess we have a question from chat. We can start from here from Baijie Xu. He's asking, is the expression of oxytocin decreased in the TRPC5 deletion mice?
Okay. Okay, that's a good question. Because oxytocin is very important for the, for example, for the milk, the release. So we also measure the oxytocin neurons, the number, and found in the PVH oxytocin neurons number is slightly decreased after the in the mutant compromise, but for the deletion, specific deletion with the fact for the compromise, we did not detect the oxytocin decrease.
Yeah, indeed, I have a follow-up question here about these oxytocin. Some of the defect these TRPC5 mutant mice. So I don't know what's their like relationship is upstream or downstream. Like, do you think oxytocin's effect is depend on TRPC5 or not depend on it?
Personally, I do think the oxytocin neuron is dependent on TRPC5.
I mean, oxytocin itself, because you have slides showing that the oxytocin treatment, right, can restore some of the defect associated with TRPC5 mutation.
Yeah, but it just only the metabolic treatments to measure the food intake and the body weight. However, we did not to test the oxytocin treatment in the behavioral, like the maternal behavior postpartum depression. So we plan to do this, but right now we still do not have those data in hand.
Okay. Yanning.
Yes, I'm here. All right. First of all, great job. I think you present a lot of data and it's very interesting. I have two quick questions. Number one is, actually, we also noticed that male have retrieving behavior. So the first question is, did you observe any deficits from your global knockout, TRPC5 knockout in male mice? They have some deficits?
Yeah, we did the male mice in the paternal behavior because male mice is totally different from female mice because male mice one important is they will attack the pups. So we measure the paternal behavior in the wild type and the TRPC5 mutant fathers when they become the fathers. So we still say the mutation in the TRPC5 also impair the paternal behaviors. It did not to retrieve the pups when they become fathers in mutant group mice.
Okay, good. And the second question is about your future plan. I think if you become independent researcher, what's your new direction from this topic? Are you gonna follow up with the future studies of this TRPC5 story?
Yeah, that's a good question. So because we did a lot of the basic the work in this paper, one important discovery is the oxytocin may become promising the compound to treat the postpartum depression. So fortunately, we have collaborate with the Dr. Sana Furuki's group and developed the oxytocin analogs. And we plan to use this oxytocin analogs to treat in our mutant group mice, as well as the division of group mice, and to say if they can treat the postpartum depression behaviors specifically in the mice. So it is one direction of this project. And another direction of this project is, as you mentioned before, we observed in the male the mutation in the mice, they showed the impairment in the paternal behaviors. So right now in my hand, I still have another project that is going on is to identify why the father mice will show the paternal behavior, but not virgin mice. So it is also related to the TRPC5 expression.
Okay, great. Yeah, congrats. I don't have any questions.
Okay,
Another question from the chat is from
yeah.
Inila. So,
why my patients also. Yeah, I have answered the last question. The TRPC5 deletion mice show the behavior change after the father. Yes, they impair the paternal behavior in fathers. For the hormone change, we measured the prolactin level in the wild type and the mutant group mice before the suckling and after suckling. In the wild type mice, compared with the baseline level, suckling could induce the prolactin release in the blood. But in the mutant group mice, there's no difference before suckling and after suckling in the prolactin level. So we just measure the prolactin change in the mothers. Yeah.
So which question you are answering about? I think that's for the question from Inila. So it's talking about why the female TRPC5 deletion exhibit depressive behaviors. So could this relate to hormonal change?
Mm.
So I think you answered this question, right?
Oh, yeah, yeah.
Oh, okay.
There's another question Elena asked. I'm curious to know if male deletion might show anything. Oh, I think you answered the question I asked earlier.
Yeah.
Elena's second question, Yongxiang already answered.
Could you explain? Yeah, I guess so. Could you explain a little bit what's happening in these mutant mice? Like why this mutant cause the decreased expression of this TRPV5? That's question one. And question two is in like postpartum depression, I think as you mentioned, there's a lot. And what's the frequency of this mutation in like human?
Okay, sorry, could you repeat the first question?
Yes, first question is about like what's happening in these mutant mice? Why the mutant resulted in decreased expression of TRPV5? And then second question is in human populations, how frequent is this mutation in women that have
Oh,
postpartum
I see.
depression?
Okay, that's two good questions. So first your question is if the mutant mice they decrease the TRPV5 expression in the brain, right?
Yes.
Yes, exactly. We stimulate the TRPV5 expressing the neurons in the PVH as well as in the active nucleus, and all of them is decreased compared with rat mice no matter in the heterozygous in the females and homozygous, also all of them are decreased the TRPV5 expression in the brains.
Yeah, I'm asking like you have a mutant, then this channel protein that it decrease this then it's like how this mutant resulted in this decrease is like resulting RNA like, decay or is it like a protein stability, things like that.
Yeah, I think it's we did not do the RNA the screening. We just did the protein the screening, and after screening the TRPV5 expressions, the neuron is decreased in the brain sections, and um, yeah, so I did not show in here the data within the TRPV5 expression and neuron. So the TRPV5 for the thing is the protein is impaired after in the mutant compromise.
Okay.
So remember your next question is how rare the mutation happen in the humans, right?
Yeah, just to see how frequent this
Yeah,
mutation.
I I did not know the exact frequency, but I want to say it's really a rare mutation we found in the humans. Also, it's very rare the mutation, but it's provided a very comprehensive the mechanism for us, the how TRPC5 affect the metabolic as well as how the TRPC5 affect the behavioral change. Also, it's rare in humans population, but it give us a lot of idea to know the TRPC5 is functioning in the brains.
Yeah, I totally agree with you. And many of these mutant is like a rare
Yeah,
mutation.
that's
And your work is pretty solid. Indeed, my two questions indeed is connected because the importance of this TRPV channel definitely is proved by your work, right? And
Mm-hmm.
then the mutant indeed is real. But in human populations, we have lots more like women suffering from this postpartum depression. Then potentially, I think the last question from Elena is also asking, we have to think about like how the TRPC5 gene is being regulated. Is it by hormones or by other kind of mechanisms? Because this is like a calcium channel, and it's a huge gene as just checked, and they have like big introns, long UTRs. So I guess they may subject to many level of regulation. And in human populations, if you have like some modifications or gene expression change of this channel, then it may have show some either severe or mild postpartum depression. So I think that's something you may want to explore as your future direction as well.
Yeah, okay, that's a good idea.
I want to jump in here.
And
That's a great point. Can you guys hear me? This is
Yeah.
yes,
Yong. So that's actually a great point. I can't help just jumping. So, yes, the prevalence of a postpartum depression is pretty high, 10, 15% of mothers. Some people even claim 30%. The mutation of TRPC5 is very rare, right? So what that means is a lot of mothers who suffer from postpartum depression, but they have functional TRPC5. And our data
yeah,
clearly indicate that TRPC5 could be activated as a target for treatment for these individuals. So um, to me, at least, the next step is to really develop a good activator for TRPC5, and which can become very useful to treat these patients and many other symptoms that we observe, like autism, obesity. Yeah, that's my point.
Hey, Yongxiang, I want
so
to follow Yung's topic. So in case you want to target TRPC5 to treat the postpartum depression, does it affect the appetite or other uh, non-related physiological function in human? So say if you activate the
Mhm.
TRPC5 in oxytocin neurons, I think you have some data to show it also regulate the body weight.
Yeah,
And in the future, if you consider to use as a treatment for depression,